Well, hello, everybody. I was waiting a minute to allow everyone to join. It's a little bit after 2 now. I'm Andrew Smith. I'm the Chief Operating Officer and Acting Chief Scientist here at Rodale Institute. And today I'm going to be presenting on our project, exploring the link between soil and human health, protein quality, and the nutraceutical amino acid ergothionine. This is our annual project update for a multidisciplinary project that we have that is funded by the Foundation for Food and Agricultural Research. And before I get started, just a few guidelines. I'm going to go ahead and present. Any questions that you have, please put them in the Q&A, not the chat. If you have any issues with the webinar or with the Zoom, please put them in the chat. We have someone in our education department that can assist you. But I probably won't see that. But at the end of the webinar, I'll leave time for question and answer, and I'll look at the Q&A and I'll answer some of your questions. Hopefully I can get to all of the questions. So before I really get started, I just want to acknowledge our partners and collaborators on this project. So these are all the collaborators, and we have several different parts of this project. One of them is looking at long-term research trials across the United States, and those are at the Rodale Institute, at West Virginia University, at North Carolina State University, and Iowa State University. And the one in North Carolina State is a collaboration between the USDA ARS. Another part of this project is looking at different wheat varieties, really looking at varieties from ancient and heritage wheat to modern wheat. And in addition to work here at Rodale Institute, we have wheat that we're growing at partner farms, and those partner farms are Lakeview Organic Grain, Kamut in Montana, Dickinson College Farm, and L&M Farms in Michigan. We also have several collaborators from USDA ARS, and our third part, or another part of this project, is looking at the soil microbiome and how that impacts crop nutrition, and more specifically, this amino acid ergothionine that I'll talk a little bit about later. And scientists at University of Delaware, Stroud-Water Research Center, and the USDA ARS are all participating in that part of the project. Today, I'm going to be specifically talking about the data at our long-term research trials. And I want you to keep in mind that this is year 1. So we will do analysis for 3 years at all of the long-term research trials. So that'll give us much more information, much more ability from a statistical perspective to analyze data and come out and have conclusions and outcomes. So let me start with why we even care about this, how we started going down this road. At Rodale Institute, one of our research priorities is solving food insecurity. And part of that is looking at the nutritional density of food, as well as looking at agricultural models that can provide access to vulnerable populations. So the FAO defines food insecurity as the lack of regular access to enough safe and nutritious, and we highlight nutritious food for normal growth and development and an active and healthy life. And some other definitions say the chronic or episodic limited access to safe, and there that is again nutrient-dense foods to support health. And I'm showing you a slide here from one of the most recent global assessments of micronutrient deficiencies. It's been a while since there's been a global assessment of micronutrient deficiencies. And you can see that a great proportion, especially of those that are in developing countries, are malnourished or have some nutrient deficiency. This study specifically looked at 2 populations, preschool age children from ages 6 to 59 months and non-pregnant women ages 15 to 49. Pregnant women are especially vulnerable and tend to be highly deficient in nutrients since have greater nutritional needs during pregnancy. So these looks at populations during development and during what would become a normal lifetime. And what I'll highlight here is if you look at the UK, the data for the UK, a developed country, you see that close to 40% of preschool age children are deficient in some or at least one nutrient, and over 50% of women aged 15 to 49 are deficient. And the gray bars here, the gray part indicates that there's deficient in more than one of these nutrients. So that's globally. If we look at the United States, we find this trend where we have approximately 70% of all Americans are overweight or obese. They have some metabolic disorder. And most Americans also are deficient in some vitamin or micronutrient. And it actually, what this graph is also showing is that as people increase their weight, they tend to have greater vulnerability to being deficient in these vitamins or minerals. So we see clearly it's not just a problem in the developing world, which maybe it's access to healthy food. It also looks at even in countries, in developed countries that have access, something about our diet, or maybe, or what we're asking the question is our food actually nutritious? And so today and this project focuses mostly on wheat, as well as corn. And the reason we chose wheat as our model crop is because it's the second most consumed grain in the world behind rice. So if we can make changes in wheat, then we can have potentially broad health impacts. And what we've seen, coming from, just kind of transitioning from that slide, is this shift in the nutritional quality of wheat over time. So this is data from the long-term broad bulk wheat trial at Rothamsted Research in the UK. So you can see from 1840 to about 1960, there was really no change in levels of the minerals that I'm showing here in zinc, iron, copper, and magnesium. As a matter of fact, what this data is showing is that using farmyard manure compared to synthetic or soluble forms of fertilizers may have actually increased some of them, like copper was increased with farmyard manure application. But in 1960, we started to see this decline in nutrients. And what that can be attributed to is a shift from traditional wheat to what are now more semi-dwarf wheat, which you can see in the top slides, increased grain yields. And so it's an example where our breeding has increased total food production, but maybe having an impact on the nutritional quality of our food. Other studies that investigated grain from about 1920 to 2002 found significant declines in protein. You can see here we have corn, sorghum, rice, wheat, and barley. Barley having approximately 50% decline in protein during that time period. And then here, as I'm showing to the right panel, not just total grain protein, but some amino acids. These are important essential amino acids that have had significant declines during that time period. And this is that study that was looking at corn. And what it's demonstrating is during that period, they looked at multiple maize varieties that were released from 1920 to 2000, and they grew them all side by side at 3 separate locations. And the trend was we see a significant increase in starch content in those over time in those varieties, and a decline in protein. So while we're having a decline in protein, we have an increase in starch. And what's also worth noting is as some of the health recommendations are to shift from a meat-based diet to a more plant-based diet, we could be eating more grains, but in order to meet our protein needs, we might be eating more grains, which have higher levels of starch, which could be leading to those obesity, overweight, and metabolic disorders that I mentioned earlier. And another alarming or something worth noting or taking a look at is how climate change might impact nutritional quality. So there's been studies that have started to look at increasing ambient CO2 uh, of the air around the crops that are grown and comparing them to the current, so maybe I call it enriched carbon dioxide versus the ambient carbon dioxide. So here's an example in Australia where they're growing wheat, and in this 12-meter circle, they're actually increasing the carbon dioxide content in the air to model what the expected greenhouse or the expected carbon dioxide levels will be in 2050. And they can compare them to the ambient air in other plots that are right next to it. So that you can see the wheat growing right behind this ring. And they're doing similar things with rice in Japan and China. So here's data from that research institute. So this is wheat, and this is all the different years. If I can get my cursor to work, there we go. If we were along this dotted line, that would indicate that there's no change in protein levels. So what they've done, to orient you, if I look at this cross here, this plus sign here, that's from 2015. Those that were grown in the regular plots under ambient air conditions, which is current today, is about 410 parts per million, had about 14% protein concentration. In those same plots that were grown within the ring with elevated carbon dioxide at 550 parts per million, had about 11% grain protein. So you can see that we're seeing this decline in grain protein, especially for wheat, as ambient CO2 levels or as CO2 levels increase. And using this data that's been replicated multiple times in multiple studies, is predicted that the C3 grains, so here wheat, rice, and barley, will have significant declines in protein. But the C4 grains like corn and sorghum may actually see an increase in grain protein. And partly what's happening is as there's increased carbon dioxide in the atmosphere, we're going to increase yields. But that may not, so we'll continue to grow higher levels of protein, but it'll be diluted in the grain. But at those temperatures, these crops are not able to assimilate nitrogen at the same level that the C4 plants. C4 grains are more tropical plants, they're more heat-loving plants. So at higher temperatures and under drought conditions, they have greater ability to assimilate nitrogen. And that's part of what we've seen, is that as we increase nitrogen or we have higher levels of nitrogen availability, we expect to see higher levels of protein in our grains. And so what we wanted to do was recognize that things are different in not only our production methods, but also in the soils and the climates where we grow our food. So I'm going to be showing specifically the data from agricultural research stations at Iowa State, West Virginia, North Carolina State, and at Rodale Institute. And let me briefly describe those research projects. So the Farming Systems Trial is the longest running side-by-side comparison of organic and conventional grain cropping systems in the United States. It's now will be entering its 43rd year. And what we have is we have conventional systems that pretty much follows the standard practices in approximately 70% of the land base or the cropland in the United States that is doing a corn and soybean crop rotation. One of our entry points, one of our plots and replications in here does have wheat in the rotation. The legume system is only using plant-based legumes as a source of fertility, and that's an organic system. And the organic manure system is organic, mimicking a livestock farm that has manure that can be composted and recycled back onto the field. And it also has a perennial phase to grow forage for those animals. In 2008, all of these systems were split into a tillage and no-till. The no-till in the conventional is continuous no-till, so there's been no tillage since 2008. And in the organics, legume and manure systems, these are actually what we call cover crop based or rotational no-till. So we're still tilling to establish small grains. We're still tilling to establish cover crops, but we're no-tilling our corn and our soybeans into winter annual cover crop that acts as a weed suppressing mulch. So that's a brief description. Iowa State is looking much more, the differences are between conventional and organic and crop rotation. So the conventional system is corn and soybean, and then there's 3 different organic systems: corn, soybean, corn with oats, and then alfalfa more as a cover crop or green manure for the following corn crop. Corn, soybean, oats. So it removes one corn in the phase, one production year of corn, and then again has alfalfa. And then the third has alfalfa for 2 years, grown as a hay crop. And this has now been 25 years that this experiment has been running. And West Virginia, it's much more looking at cropping and livestock integration. So all of the treatments are organic. And there's the crop rotation is corn, soybean, wheat, followed by a forage brassica. But where livestock are integrated, there's a orchard grass red clover phase where the livestock can graze for 3 years prior to the grain rotation. And then it's further split between high fertility, where 20 tons per acre of composted dairy manure are applied prior to corn and wheat, or no additional fertilizer is applied. So there's 4 different systems that include no livestock with high fertility and low fertility, and then livestock with high fertility and low fertility. And this is in its 23rd year now. And at the Center for Environmental Farming Systems in Goldsboro, North Carolina, there are several different research studies going on, but the one that we're specifically looking at is comparing conventional corn, which is a corn, soybean, and grain sorghum rotation, and an organic system that's corn, soybean, sunflower, with a year of cover crop and stale seedbed to improve and set up the fertility for the corn. And the organic system this year, within the data we have, is from full tillage, and the conventional system is comparing a full tillage system to a continuous no-till system. And that study, and it's been 18 years that this study has been evaluated. And then the last study that we'll look at is the ROSE experiment at Penn State University. So ROSE is Reduced Tillage Organic Systems Experiment. And this is very much set up to look at different levels of tillage in organic systems. So it's a wheat, corn, soybean rotation with 4 different levels of tillage intensity. So system 1 has moldboard plow, heavy soil disturbance rating for weed management, and the soil disturbance rating is approximately 350. So that soil disturbance rating is basically the higher, the more intense or more passes of tillage are occurring. System 2 is more shallow till. System 3 is reduced till, where they're using a cover crop based no-till approach when planting soybean. And then system 4 has a 3-year perennial phase, and therefore during that 3-year perennial phase there's no tillage, so that further reduces the tillage intensity. Before I show the results from the first year of this project, I'm showing you some of the results from a recently published paper from the farming systems trial. This is data that was collected from 2008 to 2020. During that period, since we started included the no-till treatments in the farming systems trial. You won't see anything here about tillage because most of our results found that there was no difference in grain quality between the tilled and the no-till treatments. And most of the difference that we have observed was by treatment, by organic or conventional systems. So briefly, I've highlighted the protein. What we see is in the corn manure system had the highest protein. In wheat, the conventional system had the highest protein concentration. And in soybean, the legume system had the highest protein concentration. And what we think is that this is attributed to available nitrogen. So the manure system, especially for corn, has high levels of nitrogen applied through composted dairy manure, cover crops, leguminous cover crops, and the perennial things like alfalfa. The wheat, the conventional system, actually gets 60 pounds of ammonium nitrate top dressed in the spring around this time of year, where the organic wheat gets no additional source of fertility. And in the soybeans, the legume system tends to be the lowest in nitrogen availability, but since soybeans are a leguminous crop, they don't really need additional sources of nitrogen to achieve top production. So we have a hypothesis based on a lot of our data at the Farming Systems trial, but also really looking at a lot of research and in many cases in conventional farming systems where they're applying different levels of nitrogen, that as we increase soil nitrogen availability, we should increase grain protein. But of course, we want to be careful because we don't just want to inundate the soil with synthetic forms of nitrogen that can be lost in the environment as nitrous oxide, which is a greenhouse gas, or leach into groundwater or into streams. So the form of nitrogen might be important, and we're asking the question, can we get enough nitrogen using organic methods of fertility? The other thing is a lot of our data, when we looked at, we take soil tests after crops are harvested, usually in November, and then we might look at grain nutrients, and it doesn't really tell us, what's in the soil in November or December at the end of the growing season. Doesn't really tell us what was available to the plant. So as part of this project, and all of the plots and all of the fields that are part of this project across the country, they're all in corn. We're sampling at silking, so at the vegetative phase, and at tasseling, what we are measuring is plant tissue, so we can look at the total nitrogen and the grain, and then at maturity we're measuring the grain. And I've highlighted specifically what we're going to talk about today: protein, amino acids, and ergothionine. I've eliminated minerals simply for time, and then B vitamins and calories we're still waiting to get back from the labs that we've sent off. And then at the same time that we're looking at the plant tissue testing during tasseling and silking, we measured nitrate, ammonium, which are readily available sources of nitrogen the plant can take up, right then from the soil. Total nitrogen, which could be in different forms in the soil. ACE protein, which is a form of nitrogen that is not readily available. We know that it's coming from microbes in the soil that will readily and forms that will readily break down and become available, but less likely be lost to the environment through into the atmosphere or into the groundwater. And then minerals, and we're also looking at ergothionine concentrations in the soil. And we're doing the same thing in wheat. So we're looking at heading. And so in both cases, both in corn and wheat, what we're looking at is just at the beginning, just before grain formation starts. Does the plant have sufficient nitrogen, and what forms of nitrogen, and does that in the soil and both in the leaf, and does that translate into protein, amino acids, and other nutrients in the mature grain that we're measuring? And so I'm going to start with corn, and here we're looking at the results from the Farming Systems trial at Rhode Island Institute. And some of the highlights here are that the biggest differences that we see in grain protein are between systems. So higher grain protein in the conventional system. And this may be because we fertilize at planting in the conventional system, and then we side dress later on in the season, just before stalk elongation And so there's several times that we are applying synthetic nitrogen. And you can see that translates into soil nitrate and soil ammonium being higher in the conventional system, although total soil nitrogen and soil protein, forms of nitrogen that are there will be readily available later in the season or throughout the season, were actually higher in the organic system. So we see higher protein levels in the conventional corn, and we think that this could be related to the available nitrogen levels that were sampled during silking. Now, this is from the Farming Systems Research Unit in North Carolina. And in this case, the total grain protein is higher in the organic system. Plant tissue nitrogen, so the amount of nitrogen that was accumulated prior to this taking the tissue test, was higher in the organic system, as well as soil nitrate. So again, we see a relationship between grain protein levels and available soil nitrogen. But in this case, it's completely reversed. We're seeing higher grain protein levels in the organic system and not the conventional system. And then lastly, when we look at corn, this is from the long-term agricultural research trial from Iowa State in Greenfield, Iowa. And we see no difference between the conventional and the organic as far as grain protein. I've included the reduced till. The reduced till is the one that has 2 years of alfalfa, because that's less tillage during that period. I'm not sure if it's a true, no till or reduced till, but that's where that's coming from. But we don't see any effects of tillage, and soil nitrate as well as soil protein are higher in the organic systems, but that did not translate into higher protein levels in the organic system. So I think this, this gives a good example. I guess one thing is that we saw tillage was not a significant factor related to corn grain protein. That kind of corroborates our research that we've published that didn't find a big difference in most nutrients between our till and no-till treatments in the farming systems trial. Although some of our data shows that when we use tillage, we actually increase the availability of nitrogen because we're plowing in cover crops, and we're creating increased respiration in the soil. So we sometimes see increased protein levels and nitrogen availability with tillage. But I think this also demonstrates this juxtaposition, or this, we want to say, well, is organic better, or we see organic higher, and or we see protein levels higher in organic versus conventional. But you can see we looked at 3 different locations across the country. We had really 3 different results, but they're all managed differently. They have different soil types and different climates. And this was only 1 year of data. So we're hoping that as we put 3 years of data to this, more clear results will start to fall out. But there's some trend towards higher available soil nitrogen, leading towards higher grain protein levels. And what's exciting there is that we can see that organic forms of fertility not only can increase production, but can help us maintain grain protein levels. And one thing worth mentioning is we're not necessarily looking at apples and orange, or we're not really looking at apples and apples, because in the case of the conventional, we're using GMO crops, and in the organic, we're using non-GMO, organically approved corn grain. So the varieties are not exactly equal, but we control for that with wheat. So in the wheat trials, it's the same variety in all the treatments, whether it's conventional and organic. So it's maybe a little better comparison. So the first we'll look at is the Rose experiment at Penn State, when we're looking at the wheat. And we see some treatment effect on grain protein, but it's not really, there's no clear trend that relates to tillage. Remember, the soil, or S1 was the highest tillage intensity and S4 was the least. So we're not necessarily seeing a clear trend where increased tillage or reduced tillage impacts grain protein. But we are seeing differences with the treatment, which may be related to fertility and inputs or management. This is the West Virginia organic crop and livestock farm experiment. And here again, we don't see any differences in grain protein between any of the treatments, despite the fact that the fertility treatment had significantly higher soil nitrate levels, as well as, and the livestock treatment actually looked like it had higher total soil nitrogen levels, and soil protein was higher in the livestock and from the fertility treatments, but none of those really translated into any differences between treatments in total grain protein. If we look at, this is the farming systems trial at Rodale Institute, again, we don't see any differences in grain protein between any of the systems, despite the fact that we see higher soil nitrate levels in the organic system and higher soil protein levels in the organic system. One thing to mention is this is the organic legume system, and it is the least productive. So in future years, we will have the organic manure system included in this study. Because of the crop rotation, they don't exist every year. And the organic manure system receives composted dairy manure. And so there's a strong potential that we'll see higher levels of soil available nitrogen, and potentially that will result in changes in grain protein like we saw in our 12-year study that I presented earlier. But what I recognized too was that the differences between the no-till and the till and the conventional is very different between the reduced till and till and the organic, because the no-till and conventional is complete continuous no-till. In the organic, it's reduced till, but we're still occasionally tilling. So I did some analysis where I separated the conventional and the organic apart. When I did that, I found no difference in the by tillage in the conventional system in grain protein, but in the organic system, the reduced till actually had a higher grain protein than the tilled system. And then if you look over at soil nitrate, I have a cross there, because while there was no statistical difference between the soil nitrate in the reduced till and the full till system, the p-value of 0.07 shows that there's a trend, or it's marginally significantly different. So again, potentially soil nitrate level availability is impacting total grain protein. And I wanted to take a little deeper dive into amino acids when I separated the conventional and organic. So when I looked at the conventional, I found no differences for any of the amino acids between tillage treatments. But in the organic system, I found many of the amino acids that we tested were higher in the organic no-till system. And one of them, methionine, was actually higher in the wheat grown in the organic tilled system. So it starts to tell us that management can have some impact on total grain protein, as well as different types of amino acids. But we certainly need to dig deeper and try to understand this. So it looked like in our wheat data that the tillage played a minor role in regulating wheat protein levels. Fertility management did not significantly change wheat protein levels. This could just be indicative of the fact that there was sufficient nitrogen in all of these systems to produce, adequate levels of yields, and the plants were not suffering or stressed for nitrogen. And certainly wheat requires less nitrogen than corn does, so there's probably less chance of nitrogen deficiency in wheat than there would be in corn. We were comparing the same varieties in the wheat, and one thing worth noting, or maybe taking a look at, is the variety that was used in the Rodale Farming Systems trial was the same variety used in the West Virginia crop and livestock experiment. And we see higher protein levels in the Rodale Farming Systems trial. And it's really hard to say why that is. It could be management. It also could be just we have a completely different soil type or we had different environmental and climatic conditions that led to that. But just again, it shows the variation that can exist between locations, between management. And now I want to shift to looking at ergothioneine. So if you've heard any of these presentations before, you may have learned a little bit about ergothioneine, and just for time's sake, I won't go really deep into ergothioneine because it's an extreme area of interest in the medical field right now. It's an amino acid, and it's only produced by fungi or fungi-like bacteria. It is not synthesized by plants in any way, but we know that a lot of plants have it, both in their their leaves, but also in the food that we eat. This is where we get ergothioneine from. So it's obtained through our diet. It's in plants, but we know that they can't synthesize it. So we're starting to make this connection between the health of the soil and this compound that has had extreme or an area of major interest for the medical community right now, and how we manage soil ultimately links to human health. What we do know about ergothioneine is that it's an antioxidant. It has anti-inflammatory properties. Studies that are being done suggest that it has neurodegenerative, and it's cytoprotective, and it seems to be protecting against neurodegenerative diseases and cardiovascular diseases. Humans actually have an active transporter that moves ergothioneine. So the body recognizes this compound as being important, and it moves it to areas, usually tissue that's been damaged or stressed. So that's why it indicates that it's going to be clearly important for human health. And you see that it's an amino acid, so it has nitrogen in the compound. So it could, levels of nitrogen in management could change concentrations of ergothioneine, both in the soil and in the crop. And one thing we do know is that when we till, and more frequent and intense tillage disrupts fungal populations in the soil. So if fungi in the soil is the source of ergothioneine and we are tilling, then we might be disrupting fungal populations and reducing ergothioneine in the grain. That's our hypothesis. And you can see this is from a study that was done by some of our colleagues that are part of this project, Dr. Bielman and Dr. Ritchie, who were part of this project at the Penn State tillage experiment that's overseen by Sjoerd Dijker, where they have, it's all conventionally managed, but all of the, you can see here, corn, soybean, and oats, all managed from the same way from a standpoint of fertility and varieties, but they only differ in their level of tillage. And there's we clearly can see that reducing tillage, especially in the soybean and oats, the no-till and the chisel plow treatments, which are less intense tillage than the moldboard plow, have higher ergothioneine levels than the treatments that are getting a moldboard plow or more intensive tillage. So we have a hypothesis that tillage in long-term system trials will negatively affect ergothioneine concentrations in grain. And we have some data ourselves in the farming systems trial that suggests this is true, that where we reduce tillage, in our reduced till or no-till systems, we see higher levels of ergothioneine in grains. And I presented some of that previously. So this is again from first year data, the farming systems trial. We're specifically looking at ergothioneine. So ergothioneine levels were significantly higher in the conventional grain in wheat. This was what was probably the most pronounced, was the conventional no-till system had the highest grain ergothioneine concentration. And so tillage did play a factor here. But when we look at the soil ergothioneine, we actually see that the ergothioneine levels in the organic treatments of the organic systems were higher. So there's a disconnect. And we see the same thing in corn. So if we look at corn, there was actually no statistical significant difference in ergothioneine levels in the grain, but there was higher ergothioneine levels in the soil. So this kind of goes against our prediction. We expected that tillage would impact grain ergothioneine concentrations. But we also expected that if we have practices like cover cropping and using compost in the organic system, and what we've seen in previous studies, that we can increase fungal populations and fungal biomass in organic systems, then we could increase ergothioneine concentrations in the soil, which would ultimately lead to increased ergothioneine concentrations in the grain. So part of that seems right. We see that we're increasing ergothioneine concentrations in the soil, but that's not leading to increased ergothioneine concentrations in grain. So let's take a look at other studies. Here we're looking at corn in North Carolina study and at the Iowa State University study. In North Carolina, we're not seeing any difference between the conventional and the organic or from tillage treatments on ergothioneine levels. And again, and correspondingly, we don't really see differences in the concentrations of ergothioneine in the soil. But in Iowa, we do see higher grain ergothioneine concentrations in the conventional. And again, this is despite seeing marginally significant increases in in soil ergothioneine in the organic. So again, there seems to be potentially a disconnect between grain ergothioneine and soil ergothioneine, at least in Iowa. And then when we look at wheat, we clearly what looks like a trend of higher ergothioneine in the reduced tillage treatments, or at least lower ergothioneine in the highest tillage treatment, although it's not significant. So this shows that we probably need more years. So maybe with 3 years of data, this would be a significant result. But again, they don't clearly match the amount of ergothioneine that's in the soil. And similarly, in the West Virginia trial, we're not seeing any differences in grain ergothioneine, but we are seeing higher ergothioneine concentrations in the livestock treatments, but not necessarily from the fertility, although it looks like, again, there might be a trend where the high fertility might be increasing ergothioneine. The combination of livestock and high fertility could be increasing ergothioneine. So again, more data added to that could bear out a more significant result. But again, we're not really seeing this connection between soil ergothioneine and grain ergothioneine. So I'd say that it's inconclusive, but tillage may play a role in grain ergothioneine concentrations. We expect, as we add more data, that we will see that. Fertility management may play a role in grain ergothioneine concentrations, but again, we need to add more data to that. But what's clearly we're looking at, or what we're seeing, is that the levels of soil ergothioneine do not appear to be closely associated with grain ergothioneine levels. And I'm just going to show one bit of data, which I think is really great. This is kind of hot off the press. This is from our collaborators on the project from USDA ARS in Winmore, Pennsylvania, in Wade Heller's lab, and his postdoc, Joseph Carrera, just published this probably a month or two ago. And I think this is the first time that we demonstrate a role for our buscular mycorrhizal fungi in the uptake or potentially the biosynthesis of ergothioneine. So what they've done in this study, looking at both wheat and oats, this was done in pots in a greenhouse with either sterile soil. So the sterile soil is media that has been autoclaved, and so it has no microbial life. And then we have a native soil, which was not autoclaved, but was not inoculated with any mycorrhizal fungi. So it's just natural native mycorrhizal fungi. And then they inoculated with 2 different species of mycorrhizal fungi, the S. calvospora or the C. atunicatum. And they actually did this with multiple, they did multiple crops, multiple species of mycorrhizal fungi. And they're going to have the stage later as this data becomes more mature and they can dig more into depth. But what you can see is that in the native soil, we have more than double the amount of ergothionine in both the wheat, and looks like maybe triple or quadruple the amount of ergothionine in the oat grains from a native soil compared to a sterile soil. And then one of the species, the S. calospora, when it's compared to its counterpart that was mock inoculated, we see an increase when we have inoculated the pots or the plants with that specific mycorrhizal fungi. But the other mycorrhizal fungi species did not see a significant increase. So this tells us a couple things. One, that, and I guess one other point to note here that isn't on this graph is that there was a positive correlation between mycorrhizal fungal inoculation in the roots and ergothionine concentrations in the grain. So we're clearly seeing that as you have more association with mycorrhizal fungi, we have increased ergothionine concentrations. So it's what this is basically telling us is that mycorrhizal fungi do play a role in ergothionine uptake, and it's species specific. So some, but not all, mycorrhizal fungi play that role. So clearly there's a lot more work to be done here to understand, but this could be part of the disconnect and what fungi are in the soil and how tillage or management might impact mycorrhizal fungi and other fungi that ultimately impacts the plant's ability to take ergothionine up from the soil. So a lot more work to be done, but I thank you for listening, and I'll take a look at questions. I've included my contact information if you have any questions after the presentations, but it looks like we have a little bit of time for questions, so I'll go ahead and take a look at the questions. So the first one says, is your test for grain protein actually a measure of nitrogen? If so, is a higher nitrogen grain content desirable? So there's two ways to do the grain protein, and we did both. One of them is crude protein, which is basically just measuring the total amount of nitrogen and then using a factor to multiply to that to come up with total nitrogen. The other one is a complete amino acid profile that looks at every the concentration of every amino acid and then takes the sum of that to get grain protein. What I presented today was the latter. So it was the sum of all amino acids. In some cases, there is a slight difference between the crude protein and what I call the total protein. So that's how we did it. Is a higher nitrogen grain content desirable? I don't think the data that we have here can dig into this adequately, but there is this question of, free nitrogen and nitrogen that doesn't get assimilated and turned into a protein, potentially, soluble forms, more soluble forms of fertilizer could result in the plant taking that up and not being able to convert that to protein quickly. That's a hypothesis that many have proposed. The other thing that we want to look at, you'll notice that this said the title was protein quality. And so one of the questions we have and why we're testing all the amino acids is there a difference in the amino acid profile based on types of fertilizer? So I think we need to dig in a little deeper to that. Protein quality basically is the relative proportion of essential amino acids that are digestible compared to the total protein. So that's a way that we'll look at that. We'll dig into that a little bit deeper to see if forms of fertility could result in different amino acids and protein quality. The next one is very interesting results with the ergothionine. When are soil ergothionine tests taken? At the time of seeding, at the time of harvest? It seems like soil levels would be more critical at different developmental stages. So that's a good point. So I didn't mention this, but our previous research was looking at grain ergothionine in oats and corn and wheat from our farming systems trial. And then we said, let's take a look at the soil. And the soil was always taken in November, usually after harvest. And that's why this project's taken a little closer look at both nitrogen and ergothionine levels just at the time of grain development in the field, because perhaps, like you said in the question, the ergothionine levels in November have nothing to do with the ergothionine levels, or maybe they're completely different than the ergothionine levels during the growing season when the plant can actually take it up. Maybe it would be great to do it on a weekly basis or bi-weekly basis, but partly because of funding, because we're doing this broadly across multiple sites, we're only doing it one time, which is at that heading phase or the grain development phase for both corn and wheat. So how many samples are you testing in each system? So in the long-term trials, all of these are randomized and replicated. So for instance, the farming systems trial, we have 72 plots. All of the treatments have at least 4 replicates. So we are sampling in every one of those plots. And that's true they might be a little bit different design in all the other long-term research trials, but they're all randomized and replicated. So we're testing multiple replicates. And then in the lab, they may do triplicates, for example. So when they're looking at ergothionine or total nitrogen and we send them soil or plant samples, they may be doing triplicate samples and then giving us an average of that triplicate. So I hope that helps. And then when we test for soil, we're taking usually a minimum of 10 soil samples throughout a plot and then aggregating that together as a representative composite sample that we send off. And same thing when we do plant tissue and grain, we're taking random samples from throughout the plot and then creating a single measurement. So what is mock inoculated in the ergothionine experiment? So it's a little bit confusing, but basically it's completely native soil. I'm sorry, it's completely sterile soil. And then the addition of so we want to remove the potential addition of the media that comes along with the addition of the mycorrhizal fungi that's inoculated. So it's basically adding media that has no mycorrhizal fungi and is completely sterile. I don't know if that makes sense. What might make sense is like if we wanted to spray a fertilizer, we mix the fertilizer with water and then spray it on one plot, and then we actually spray just water on the other plot because maybe adding water had some treatment effect that we didn't think of. So that's kind of why it's called a mock inoculated, because it's inoculated with something that's added, but it's what's added does not have any of the mycorrhizal fungi. I hope that explains it. Wade and Joe usually describe it a little better than I do because they do it so often and describe it so often. Do we know why fungi produce ergothionine and then provide it to the plants? Very good question. And I don't know if we do completely know, but it looks like it probably has something to do with oxidative stress, which might be why it is a antioxidant. And we don't know a lot about plants. So in humans, like I mentioned, we see that it's an active transporter. So we actually have, and I'm not a medical scientist, but basically the human body recognizes it and it's not passive. It doesn't just go into our blood and then just go. It actually, our body takes it and moves it through cells to get it where it needs to be. We don't know anything about that in plants, whether plants have some transport mechanism to move it to different places where it wants is. That would help us understand why, if it moved to different parts of the plant. So I'd probably say no, we don't know that much about why ergothionine exists. Which labs are you using for testing? So we are sending our plant, make sure I get this all right. Well, let's see. So University of Missouri does amino acids and the protein. All of our ergothionine is done with our collaborators in Hershey, Penn State Hershey Medical Center. The soil samples are sent to Penn State. The plant tissue samples, I believe, are also sent to Penn State. and we are sending vitamins and what am I missing? Vitamins and our proximites, which includes carbohydrates and calories, which is something, this is data we haven't gotten back yet. We're sending that to Eurofins Scientific. And so we're waiting to get that data back, which is why I didn't present it. The other question was, given the seemingly higher response in oats to ergothionine and the soil fungi, do you have any plans to add oats to the trial? So it's clearly recognized that oats has high levels of ergothionine. Before we even looked at the management practices and how they affect ergothionine, there was some publications, I think just one, honestly, there might be one more now since then, that showed higher ergothionine in oats and higher ergothionine in black turtle beans. And so if you dug into the paper that from Joe Carrera that I mentioned, you'll see that he used black beans and oats, or some literature that suggests that asparagus was high in ergothionine, so they tested asparagus. And the only reason we went with wheat over oats, I mean, oats it's actually an easier plant to grow in the greenhouse, but for total global consumption, it doesn't come close to wheat. So that's the reason we chose wheat. But we're, a lot of our greenhouse and our our controlled studies are using wheat. The other thing is, if you look at all those research trials, the farming systems trial doesn't have oats in the conventional. So we don't have we have a comparison between the legume and the manure system, but not in the conventional. A lot of the other long-term trials aren't necessarily growing oats. I don't think the Rose did. I don't think the North Carolina study was. So it's it's not as widely grown as wheat also. So those are some of the reasons why we chose wheat. But if you ask me, hey, if I was going to change my diet to get more ergothionine in my diet, I would recommend eating more oats because it's a high source of ergothionine. And I didn't mention it, but maybe it's just intuitive, but mushrooms have high levels of ergothionine, and different species of mushrooms have different levels. So I don't want to spend too much time on that, and I'm probably not the expert in that, but mushrooms, oats, black beans are all really high sources of ergothionine that could be for our diet. So I don't see any other questions in the Q&A section. So I thank you for joining today. I'll take a quick look in the chat. Thank you, Caroline, but no questions. And I'll go ahead and we'll end the webinar there. Thank you all for joining us today, and have a good day.